What if the ability to repair a spinal cord injury became a reality in the next few years? The Spinal Injury Project here at Griffith University is on the cusp of making this a reality with a world-first cell transplantation and rehabilitation human clinical trial. Today we're going to take you on the inside and show you our laboratory. So sit back and enjoy our virtual tour.
Hi, and welcome to Station 1. I'm Francesca, but you can call me Fran, and I'm a PhD student here at the Clem Jones Center. So today I'm going to give you a little bit of a background about who we are. So as you can see here at my back, we are a really big team. We count over 30 researchers. We work in 3 different projects, but today we're going to focus on the Spinal Cord Injury Project. We have lab leaders, research fellows, research assistants, students, both master and PhD students like me. Also, what we would like to show you today is that our biggest strength is our diversity. We come from 14 different countries. And this gives us so many different backgrounds, which means a lot of different ideas. That's our little secret recipe for our projects. Now let's move here. So it's just not about people and about our ideas and about the projects, because without the people that support us, we wouldn't be able to be here. As you can see here, the trees and the fruits are alive just because they have water. And the water that keeps us alive comes from our funders, people that support us, like the Perry Cross Foundation, of everyone through the Perry Cross Foundation that can reach us. All right, now we can move to some more about the real context of what we do. So what we're trying to do here is to develop a cell-based transplantation therapy, which is going to go to people affected by spinal cord injury. So our main goal is to develop something for them. That's why we really focus on consumer engagement. We currently have live a survey where we're asking people with spinal cord injury, what do you want? What do you expect from a therapy? Here you can see some of the preliminary data that we have collected from it. And you can also scan the QR code and fill out the survey today. That was it for me. So now we're going to move into the lab and we're going to listen to station number 2 with our Professor James St. John. Let's go in.
Hi, welcome to Station 2. I'm going to tell you more about our therapy, so come on and see what we're doing. I'm Professor James St. John. We have 3 main projects: the Spinal Injury Project, the Peripheral Nerve Injury Project, and Neurodegeneration. Our biggest project is, of course, the Spinal Injury Project. Spinal cord injury is a devastating injury because it happens to normal people doing their normal daily activity, such as driving a car, riding a bike, or playing sport. 21,000 Australians are affected by spinal cord injury, and every year another 300 people join that list. Currently, there is no treatment. It's an expensive injury to Australian society, and it costs over $3.7 billion a year in socio-economic costs. But when we think about spinal cord injury, it's more than just not being able to move. It's also not being able to feel and not having control over bowel, bladder, and sexual function. So we need a therapy that can repair all those aspects of spinal cord injury. We're working on a therapy that uses cells taken from the nose. This has a long history of research from around the world. In the 1990s, these cells were first reported in the scientific literature. And in 1998, it was shown that these cells can be taken from the nose and put into the spinal cord to repair spinal cord injury in rats. Then in 2002, the world's first blinded human clinical trial, led by Queensland's own Alan Mackay-Sim, showed that using these cells was safe for use in humans. And then because of his work, people from around the world continue to work on this therapy. In 2012, a study from the University of Cambridge showed it can work in dogs. In 2014, it showed it can work in a human. This is an amazing moment in science because it showed that repairing the spinal cord injury was possible. We came into this program in around about 2015 when the Clem Jones Centre was started. And we looked at the therapy in its current state and worked out what did we need to improve to make it better, to make it more effective in more people. And what we've done is invent a powerful technology that we call nerve bridges. And you'll see more about these in the coming tour. And now we're on the verge of going to human phase 1 clinical trial. It's been a long journey. As I said, from 2016, we've had support as the Clem Jones Centre, but the Perry Cross Spinal Research Foundation has supported this research from even way before then. But it's expensive to get to clinical trial. We are now designing and scoping the trial, and we need another $8.5 million to get to a phase 1 clinical trial. It's a lot of money, but compared to the cost of spinal cord injury to the Australian Society, it's less than one day's cost. To do our work, we have a fantastic team that Fran told you about before. It's about diversity and new ideas and new skills. Some people in our team are high flyers, such as Dr. Mo Chen, who won the Research Australia Discovery Award in 2020-2021. He's the inventor of the nerve bridges. So what is this therapy? What are the cells? Well, we take cells from inside the nose. Why? Because every time we breathe in, we're breathing in air and bacteria, viruses and toxic chemicals. We know that COVID can go up your nose and cause you to lose your sense of smell. That's because the nerve cells that detect odours are directly exposed to the air, and they can die from those bacteria and viruses and toxic chemicals. But luckily, stem cells are present there that replace those dead cells every single day. Every day, about 1 to 3% of your nerve cells in your sense of smell die, but they're replaced. And it continually repairs and grows up connections into the brain. And it is the special cells called olfactory and sheathing cells that help in this repair. And so that's why scientists thought, oh, if we can take these from the nose and put them elsewhere, can we repair other parts of the nervous system? And this is what Professor Adam Akaisim did back in 2002. He tested whether taking these cells from the nose and putting them into the spinal cord of people who got spinal cord injury was safe. His work showed that it was, and for this, he got the 2017 Australian of the Year prize. The real moment in science was when, in 2014, the Polish team joined up with the University College London team to show that this can work in human. This guy here, Derek, had been stabbed in the back with a knife that completely severed his spinal cord apart from a small lateral connection. They took these cells from his olfactory sense of smell and transplanted them into his spinal cord. And after intensive rehabilitation therapy, he regained the ability to move and feel his legs. I should say, he'd been injured for several years before the transplantation. So this shows it can work. Other people in the trial didn't get such good results, so we need to make this therapy more effective for a wider range of injuries. So we come into this with our invention using a technique that we call BioBuilder. We can make large, complex cellular nerve bridges. It's fast, it's cheap, and it's effective. Importantly, these stable nerve bridges allow the surgeons to place the cells directly into the injury site where they can maximize their location to where they're needed, and also they have stable connections that allows the cells to survive. We've got, in addition to Mo Chen's Research Australia Award, the National Health and Medical Research Council Marshall and Warren Innovation Award in 2019 for the potential of this therapy design. So how do these cells work? This is a video of ants across across a gap. And you can imagine in the spinal cord there's a gap and we put our cells in. Each individual cell might have to fight for its survival, and on its own it can't do it. It's only when they work as a team that they can bridge that gap. And by putting that nerve bridge in right at the very start, we can accelerate that process. And so you can see here with the ants, they are forming this bridge. And some are forming the bridge while others are crossing over it. It's the same in the spinal cord. We have a permissive bridge that allows those nerve cells to grow across along with other cells. That's what we're creating with our cells. I like to think it's complex, but really it's quite simple. And simple is often the best way to get an effective therapy. So here's some examples of mice that have been treated with our nerve bridges. Now, we can induce spinal cord injury and the mice would normally be paralyzed. After transplantation, they can regain some function. And you'll see here, the mouse on the left, after having these cells transplanted, has regained the ability to move its hind legs, particularly the right leg. And the mouse on the right you can see with a pinch test, it's a light pinch test to the foot, and it can feel and also move. So we've restored sensory and motor function as well. And also our mice can restore bowel and bladder function too. So we can restore motor, sensory and autonomic functions. So now we're at the point, we've done the animal work and now we want to go to human clinical trial. So our therapy looks like this. We will have people who've got chronic spinal cord injury at least 12 months after injury. They will do a period of prehabilitation, so exercise-based rehabilitation before the transplantation. During that time, we'll take a biopsy from their nose, purify the cells, make the cellular nerve bridges, and then we'll transplant them into the spinal cord. That's with open surgery, so the surgeons can see that injury site and know exactly where to place the nerve bridges. And then people will continue on with rehabilitation for another 8 months after that. And then once the cells are put into the spinal cord, they work in a way that allows those nerve cells to repair. And so this is an example of a schematic of a spinal cord. And you can imagine it's been got a gap in the spinal cord. The cells there, the nerve cells, do want to grow across that gap, but they can't because of the physical cavity there, but also because there's scar tissue formed by the astrocytes. And so the neurons coming down and interneurons all around, they want to grow, but they're prevented from doing so by the scar. So we can come in with our cells, which are injected as a nerve bridge into the injury site. They'll then migrate out of that nerve bridge and integrate with the host tissue. And then once they've integrated and formed that bridge across that tissue, they can merge within the scar and allow those nerve cells that want to grow across to come across and then form connections. And then with long-term rehabilitation, the body can make sense of those connections and hopefully restore function. That's enough for me talking. Go on to the next station, you'll learn about the cells and then the nerve bridges.
Hello everyone, welcome to the Cell Purification Station number 3. My name is Mariam Murtaza. I'm a research fellow working at the Clem Jones Center for Neurobiology and Stem Cell Research for the past 7 years, specifically on the spinal injury project. So today I want to tell you a bit more about the cells that we work with, the olfactory ensheathing cells. Did you know that we lose the olfactory sensory neurons every 2 months? So how come we don't lose our sense of smell? It's because these cells regenerate, and this regeneration is helped by the olfactory ensheathing cells. And because of this reason, these cells have been used extensively for clinical trials, and they have shown success, but with some variability. But in addition to these neuronal regenerative properties, these cells have the unique capacity to work like a vacuum cleaner, where they can clean up debris at the injury site. They also can secrete interesting molecules, and they can interact with the cells like the astrocytes, which are present at the glial skull. And in the lab, we work with 2 models of these cells. So the first model is the mouse model, where we've done extensive preclinical work. And the second one is the human model, where we have volunteers who very generously donate a sample of their olfactory tissue, and we process these samples. So the advantage that we have with the mouse samples is that they are fluorescent. But you and I, we are not fluorescent, which makes it much more challenging to work with these cells. So we need specific markers to try and identify these cells in the human samples. So regardless of whether the samples are mouse or human, what we need is unambiguous identification of these cells. And we have worked extensively to try and establish a panel of markers. So when you start off with the samples, you end up with a mixed proportion of cells. And you can see that in some of the slides, where our cells of interest are red. And our objective is to then get an enriched red population of cells. And conventionally, these cells are grown flat in a cell surface, like a flat sheet. But unfortunately, in the body, it's not a flat sheet. So we try and mimic some of these conditions in a 3-dimensional format, which is much closer to what the cells look like in our body. And we want to try and minimize the precious samples that we use, so we can work with a plate such as this. So instead of working with 1 sample, we can work with 96 samples at a time, and we can get a lot of information from this. So this is an example where we can actually grow the cells and actually monitor them in real time. And we can do interesting assays such as this example here. The olfactory cells are in red and they're interacting beautifully with these neurons. And we can do similar sort of assays with the human cells where they are in a three-dimensional format, and you can see the cells migrating out from the spheroid. And we can then stain these cells with specific markers to identify the olfactory cells of interest. So a lot of work has gone, I think, into the preclinical work, but we are now at an exciting point where we are moving towards developing clinical grade cells. So the preclinical work can happen in a lab such as ours, but for the clinical purposes, we need to work at a GMP, which is a good manufacturing facility. And this is what we are planning to do, where we will do a pilot production of our human sample processing and all the assays that we have developed and validated so far in a GMP facility in Brisbane. So we are systematically working through all the different criteria that are required for us to get the approval of a clinical grade product. And what do I mean by that? So we are working to define the identity of the cell, the identity of the starting material and the identity of the final product. And we are defining the purity of the population that we are working with and the contaminating cell types that are present. And finally, we are going a step beyond what has been shown previously, where we are predetermining the functional capacity of the cells that we are working with. And of course, safety comes first. So we are going through rigorous safety testing as well to ensure that the sample is devoid of any contaminants. And this whole process of working towards a clinical grade cell product is quite complex. As you can see in this example, there is a lot of factors involved, and we are working through the tissue procurement to the final stages of the product and the logistics and all of it. And what we are hoping is that by the end of this, we will have established standard bioassays for developing clinical grade olfactory cells. And what this would do is it would provide a reference point for other groups around the world, so they can then produce standardized cell populations and preparations of the highest therapeutic potential. And what this means is that it makes our cell therapy more widespread for adoption across the world. Thank you for your time. Now we move on to Station 4.
Hi everyone, welcome to 3D Station. I'm the host of the station. My name is Dr. Ju Jin, and this is our co-host, Dr. Mo Chen. We're going to host this station together. You might have a basic understanding of how olfactory shielding cells, our magic OEC cells, could be used as a strong candidate to treat spinal cord injuries. So at this station, we'll talk about more like a technique basis, which means by means of bioengineering method, what we're going to do with these OEC cells to enhance their properties with their therapeutic potential to treat spinal cord injuries. So you can see there are a few things listed here. That's what things we want to share with you today. So we want to go through every single one, 3D printers, 3D nerve bridges, and we also share some experimental data with you. So let's welcome Dr. Mo to help you with this.
So we're familiar with all different kind of 3D printing technique. For example, we're familiar directly printing cells. Here is a machine can directly printing cells. And we also have our special technique to making 3D culture. So on the surface of cells, there are a lot of small hands. Cells using these small hands to build cell-cell interaction. This is how cells build nature interaction to build the tissue and build the human body. We're using this concept to design our nerve bridge. For example, this contains 10 million human cells, and it's only health cells. It's no any artificial material. And also, we can control the time of making this nerve bridge. For example, this one is only using 6 hours to forming. And we also can control the shapes of our nerve bridge. For example, this one is identical to the trigeminal nerve on human face. So another example is the chicken nerves. So on the right side is a real chicken nerve we get from the supermarket, and in the middle is our nerve bridge. So for getting this one, we did some 3D scanning of the real nerve and get all the information we need, and we can starting design the shape of our nerve bridge.
We are thrilled to share with you some of our experimental data, and you can see all the staining here. So we did a 3D printing of this 3D chamber, and based on that, we designed a spinal cord injury model. So you can see it here. There's a huge gap in the middle that indicate like spinal cord injuries has a gap in the middle, and we put the sensory neurons on the side, and they are growing freely, but they couldn't see or detect each other because of the gap. However, when we put the OEC, olfactory incising cells, nerve bridges in it, you can see a very clear direction towards the OEC cells. Not only the volume, but also the direction are huge, significantly different here. That's why we are very confident with our products and our nerve bridges. So understanding all these mechanisms inside of how olfactory incising cells could help with neural regeneration today might help us to prevent or even help to treat the spinal cord injuries tomorrow. Thank you.
The spinal cord is a very important part of the central nervous system that connects the brain to the rest of the body. As such, it is covered within these vertebral bones, which makes it very well protected naturally. However, injuries do happen. So I'll talk a little bit more about how the spinal cord injuries happen. This is a model of the spinal cord. It is about this thick and this wide at the thoracic level, and it has these transparent coverings with the red squiggly lines. They mean the blood vessels. The important part to remember about spinal cord is the consistency of the tissue. The spinal cord naturally is very fragile, and it's more like toothpaste. So you can imagine something like this would happen when there is an injury. The spinal cord tissue will get impacted, the blood vessels break, and the blood pools at the injury site. And every time the blood pools, the blood eventually clots, like so. And the clotted blood will resolve eventually, and you end up with a lot of scarring. Now, the consistency of this scar tissue is more like leather or fabric. So this toothpaste-like neuronal tissue from the spinal cord cannot permeate through this fabric-like scar tissue naturally and heal itself, which is why surgical intervention is required. Now, the clinical trials that have used the cells so far, they had to put the cells in a suspension form, take them in a syringe, and inject that around the injury site like so. Now, imagine putting a bit of water on toothpaste, it flows away, and that's what was observed. Most of the cells, around 99% of the cells, would not survive at this injury site after 1 week of transplantation. And this is why these nerve bridges are so amazing, because we can handle them surgically, we can place them manually right where they need to go within the injury site and make sure that they stay secure. And with this, we have observed that the cell survival has increased by a factor of 25 to 30 times what other people have reported. Increased cell survival also means that we can now predict what outcomes we are going to see more reliably and more confidently. So if you look at this image here, it's about 30 micron thick section. It's about half a hair strand thick section of the spinal cord. You're looking at the spinal cord in this and this direction, and this gap here represents the injury site. Now, the red cells that you see here, they are the OECs, the surviving OECs from the transplanted nerve bridges, and you can see that after 1 week of transplantation, there are a lot of cells surviving here. At the same time, if we just looked at the neuronal tissue and the spinal cord tissue, you'll see that there is so much debris here. This dot-like signal coming in red color is the neuronal dead cells. And because of the dead cells and the scar tissue, the natural regeneration cannot happen. However, 1 week after transplantation, you can see that the debris field is clearing out. This means that the OECs that are surviving in the injury site, they are taking up the debris. 2 more weeks after that, you can see that the nerve cells are starting to make connections. You can see these bundles traveling up here and traveling down here, right where the OECs were surviving in the previous images. And amazingly, after 5 to 6 weeks of transplantation, the wide, this big injury site has essentially shrunken down to about 1/4 of its original size. And if you look at the color within the regenerated area, it matches with the spinal cord tissue around it, which means that this is true tissue regeneration. This is not scarring, or this is not functionless tissue that has regenerated. This is real functional tissue, and we can corroborate that with the behavioral tests that we are doing. We have also developed behavioral tests that have reverse translated from the clinics, meaning that the tests that the doctors use on spinal cord injury patients, we are able to use similar tests on our test animals and establish realistic clinical expectations based on our efficacy results. Thank you.
Hi, my name is Dr. Megha Shah. I'm a medical doctor. I specialize in human pathology, and today I would like to talk to you about what's going to happen after endpoint, after the euthanasia, once we receive the tissue. What happens to that? So once we have the spinal cord tissue, we process them. It takes around 3 to 4 weeks for the processing, and then we use a machine called cryostat and cut the spinal cord into 30 microns thick sections. So 30 microns is even very thinner than the thickness of our hair. And as you can see here, there are 5 sections of a mouse spinal cord. And after that, we use this microscope. This microscope is funded by Perry Cross Spinal Research Foundation. We use this microscope to scan all these slides. And then we stain these slides with different dyes, different antibodies, so that we can see neurons and all those supporting cells. And we can see what's exactly happening on the cellular level. Are the cells that we transplanted, are they talking to the neurons? Are they talking to each other? Are the cells helping neurons in order to regenerate? So this is an example of an image that we took from this microscope. This is the spinal cord. The red bit that you are seeing is stained with a marker that stains astrocytes. So all the red are the supporting cells, and where you can see the break in continuity, that's the spinal cord injury. And the yellow cells, yellow bits that you are seeing, those are the cells that we transplanted. And after we have these slides, we take them into different software and analyze them further. So here is an example. Again, the yellow that you see are the cells that we transplanted, and white bits that you are seeing are axons. So here you see not only axons and our transplanted cells are in the vicinity, but as we examine closer on the cross section, here, as you can see, that our transplanted cells are actually wrapping and sheathing axons and possibly helping them to regenerate or to repair. So these are the findings that we are interested in. And after we get these findings, we always correlate our histological cellular findings with the motor findings. Thank you for joining me at the Station 6. And now I'll hand over to Yasmin, Station 7. Thank you.
This is Station 7. My name's Yasmin, and I'm one of the researchers here at the Clem Jones Institute. And I'm going to talk about the second component of our therapy. So the first component is the transplantation of cells, and the second component is the intensive and long-term rehabilitation program, which is presented both before the therapy, or before the transplantation, as prehabilitation, which really primes the body for the uptake of the cells, and after the transplantation, which ensures the cells are making their right connections. So this program happens on site at Making Strides. So this is a specialized neuro rehab facility that specializes with individuals with differing levels of injuries, differing age groups and things like that. And we've actually already run our first human clinical trial, which tests the rehabilitation program. And when we advertised this program, we had over 100 different applicants. And from this, we independently chose 5 individuals with different demographics, different levels of injuries and ages and things like that. And we started with a 16-week on-site program that went for 2.5 hours of exercise a day for 5 days a week for 16 weeks. And then we followed with an at-home program, which was a slightly lesser intensity that only went for an hour, again for 5 days a week for 10 weeks this time.
We're also running our second rehabilitation trial, so this is testing the pre-habilitation program. And again, when we advertised the program, we had many applicants. We again chose 5 individuals, but this time we went for individuals with a wider geographical area. We also looked to target individuals with less experience with intense rehab. And this time we also chose to film the participants, not only to show the individuals their progression over time, but also their family and friends, their clinicians and surrounding community. Thank you for listening to my section. We'll now have Andrew introducing the future of our centre.
Thanks, Yasmin. Hi, my name's Dr. Andrew Rayfield. I'm a Research Fellow and Research Manager for the Clem Jones Centre for Neurobiology and Stem Cell Research. I'm going to talk to you about the future of what the Clem Jones Centre will be and what we're doing with our full cell therapy trial. The overall process for the trial is straightforward. Take a group of people through prehabilitation to maximise their potential for recovery, as well as getting them suited to an intensive rehabilitation program. We're aiming for a 12-week program. Take their nasal biopsy, which will be prepared in partnership with a dedicated good manufacturing practice facility in Brisbane, QGen Cell Therapeutics at QMR Berghofer. There they will maximise the number of cells and assemble them into 3D nerve bridges ready for transplantation. We aim to conduct the surgery at the Gold Coast University Hospital, where we are assembling a team of surgeons and other professionals to be involved. Post-surgery, the participant will undertake intensive exercise rehabilitation for 32 weeks to help stimulate the nerve cells, to find their appropriate targets, to re-establish and strengthen functional neuronal connections. The patient profile will be patients with chronic spinal cord injury, that is, greater than 12 months since their injury. We're looking at Asia A, B, and maybe C, and thoracic to low-level injury, plus other inclusion-exclusion criteria. We are aiming for 10 patients recruited from the Southeast Queensland, Northern New South Wales area. Why haven't we already started? We have the know-how, we have the science, we don't have the money. Graciously, the Perry Cross Spinal Research Foundation has donated $2 million towards our full cell therapy trial. Other contributors have been the Clem Jones Foundation, Griffith University, and Nicola and Andrew Forrest, which have contributed $1 million each. We still have about $3.5 million to go for the full trial. In addition, we have received funding from the Perry Cross Spinal Research Foundation to scope the clinical trial and talk to regulatory authorities. We've also been lucky to receive funding from Griffith Institute for Drug Discovery at Griffith Nathan Campus and a Therapeutic Innovations Australia voucher for $30,000 to scope and pilot the cell therapy at QMR Berghofer, QGen. This isn't the end of our research. We will continue to develop our research for spinal cord injury. We will work with community, clinicians, industry, and regulators. We want to make a product that the community wants that also fits with what the industry and regulators need. What we're doing here is version 1.0. We're going to have other iterations to our therapy, version 2.0, version 3.0. After all, the first rocket built didn't get men to the moon. We've barely scratched the surface of the nerve bridge technology. We're also looking at top-up therapies using OEC cell top-ups. We're working with collaborators interstate to look at electrical stimulation of OECs. We're repurposing old drugs to make OECs and neurons more attractive to each other. We're also looking at growth factors that can show, when they're applied to the injury site, greater regeneration. The lessons we have learned here will help us fast-track new innovation and new clinical trials into the future to help with spinal cord injury, peripheral nerve injury, and brain injury, to create cell therapies to treat injuries of the nervous system. That's all from me today. Thank you for joining us, and thanks from everyone at the Clem Jones Centre for Neurobiology and Stem Cell Research to you for joining us on this lab tour. The future is bright, and we're really excited to share our progress with you. See you all very soon.